Ever wondered how scientists measure the amount of a specific substance in a solution? Meet amperometry. This is not just a fancy word, but a powerful tool in analytical chemistry that helps us understand the nature of certain substances in solutions. So what's the magic behind it? Amperometry is based on the principle of electrochemistry, where it measures the current in a solution due to the oxidation or reduction of an electroactive species. Picture this: you have a solution, and you want to know how much of a certain substance is present. Amperometry can help with that. It uses a process called Amperometric Titration, which involves adding a reagent to the solution and measuring the current that's generated. This current is directly proportional to the amount of substance in the solution. Astonishing, isn't it? But wait, there's more to amperometry than meets the eye. Stay tuned as we delve deeper into the fascinating world of amperometry. Now that we've briefly introduced amperometry, let's explore its underlying theory. Amperometry is a branch of electrochemistry, and its essence lies in the measurement of electric current. But how does it do that? The theory of amperometry is akin to a dance between electricity and chemistry. It's all about how a substance in a solution interacts with electricity. When an electric potential or voltage is applied to a solution, it urges the substance to participate in an electrochemical reaction. This reaction generates an electric current. It's this current that amperometry measures. Think of it as a conversation. The voltage is like a question posed to the substance in the solution. The electric current is the response. The strength of this response, the intensity of the electric current, is then measured and recorded. Amperometry operates under a fixed potential. This means that the voltage applied to the solution remains constant throughout the process. This constant potential is what allows the reaction to proceed unimpeded and for the current to be measured accurately. The result is a plot of current versus time, often referred to as an amperogram. This amperogram can reveal a wealth of information about the substance in question. It can tell us about its concentration in the solution, its reaction rate, and even its diffusion properties. But the process doesn't stop there. Amperometry also involves the use of electrodes. There are 3 types of electrodes typically used in amperometry. The working electrode, where the reaction of interest occurs. The reference electrode, which maintains a stable and known potential. And the auxiliary or counter electrode, which balances the current flowing through the working electrode. In essence, amperometry is a method of probing the unseen, a way of asking questions to substances in solution and listening to their electrical responses. It's a complex dance of electricity and chemistry that allows us to glean information about substances that might otherwise remain hidden. The theory may seem complex, but it's the backbone of amperometry. With the theory in place, let's now understand the principle that governs amperometry. At the heart of amperometry lies the principle that current, when passed through a solution, can be used to measure the amount of a substance present. This principle is based on Faraday's laws of electrolysis, which state that the amount of a substance produced at an electrode during electrolysis is directly proportional to the quantity of electricity passed through the solution. In simpler terms, this means that the more of a substance we have in our solution, the more current we'll see passing through it. This is because each molecule of the substance can carry a certain amount of charge, and when we apply a voltage, these molecules move, creating a current. So, if we have a solution with a large amount of a certain substance and we apply a voltage, we'll see a large current. Conversely, if we have a solution with a small amount of that same substance, we'll see a smaller current. This principle allows us to measure the amount of the substance in the solution by simply measuring the current passing through it. This is the essence of amperometry. It's a method of quantitative analysis based on the measurement of current. But it's not just about measuring the amount of a substance. The principle of amperometry also allows us to identify the substance. This is because each substance carries a unique amount of charge, which means it will create a unique current when a voltage is applied. By comparing the current we measure to known values, we can identify the substance in the solution. Amperometry, in essence, is a tool that allows us to both quantify and identify substances in a solution using the principles of electrolysis and current measurement. It's a powerful technique with a wide range of applications, from environmental monitoring to medical diagnostics. The principle of amperometry, just like its theory, is integral to its functionality. One of the most common applications of amperometry is in titration. Let's see how. Amperometric titration, an analytical method, has a unique charm. It's like a detective story where our protagonist, the titrant, gradually reveals the secrets hidden in a solution. But instead of a magnifying glass, our detective uses a measuring instrument to detect changes in current. In a nutshell, amperometric titration involves the gradual addition of a reagent, the titrant, to a solution until a reaction is complete. This process is akin to filling a glass with water drop by drop until it's full. The beauty of this method is that it can tell us precisely when the reaction is complete, or in other words, when the glass is full. It's the precision of this method that makes it truly fascinating. But how does it do that? Well, it's all about the dance of electrons. As the titrant is added to the solution, it causes a redox reaction, which involves the transfer of electrons. This transfer generates an electric current, which our measuring instrument detects and records. As the reaction progresses, the current changes, and when the reaction is complete, the current reaches a steady state. Now, where does amperometric titration really shine? It's in the detection of endpoints in redox reactions. An endpoint in a titration is like the climax in our detective story. It's the point where the reaction is complete and all the secrets have been unveiled. Detecting this point with precision is crucial, and that's where amperometric titration excels. One of the key applications of amperometric titration is in the analysis of water and wastewater. It's used to determine the amount of certain substances, such as chlorine in the water. It's also used in the pharmaceutical industry to analyze drug components. So there you have it. Amperometric titration, a simple yet powerful tool in the hands of chemists. It's a method that combines the thrill of a detective story with the precision of science, and it's a key player in our quest to understand the world around us. Amperometric titration, a simple yet powerful tool in the hands of chemists. Scene script. Like any other technique, amperometry has its pros and cons. Let's explore them. One of the significant advantages of amperometry is its sensitivity. It can detect minute changes in current, making it an excellent tool for determining low concentrations of various substances. Additionally, it's a robust method, unaffected by color, turbidity, or viscosity of the sample. Amperometry also provides rapid and real-time results, a feature that's particularly useful in monitoring dynamic processes. However, it's only fair that we also discuss the disadvantages. Amperometry can be sensitive to environmental changes such as temperature or pressure, which may affect the accuracy of the results. It's also susceptible to interference from other electroactive species present in the sample. Furthermore, it requires careful calibration and maintenance of the electrodes, which could be considered a disadvantage in terms of time and resources. Now let's delve into the fascinating world of amperometry applications. It finds extensive use in environmental monitoring. For instance, it aids in detecting pollutants in water or air, providing crucial data for environmental protection efforts. In the food industry, amperometry plays a vital role in quality control. It helps determine the presence of additives, contaminants, or even the freshness of the product. In the realm of clinical analysis, it shines brightly. It's used in glucose meters, a crucial tool for managing diabetes. Amperometry also assists in detecting other biomolecules such as cholesterol or lactate in blood samples, contributing significantly to diagnostics and medical research. From ensuring the water we drink is safe to keeping a check on our health, amperometry has wide-ranging applications. It's a testament to the versatility and importance of this method in various fields. Despite its limitations, amperometry's wide range of applications make it an indispensable tool in analytical chemistry. Now let's take a brief look at a unique variant of amperometric titration, the Biamperometric Titration. This technique is a bit of a game changer in the world of amperometry. It involves the use of not one but two identical working electrodes. You might wonder why? Well, it's all about improving the precision of endpoint detection. In a nutshell, Biamperometric titration works on the principle of maintaining an identical potential at both electrodes. When the titration reaches its endpoint, the potential difference vanishes. This disappearance can be accurately detected because we're working with two electrodes. The result? More precise, more accurate measurements. It's like having a double-check system in place. making sure that our titration results are as accurate as they can be. This distinct feature of biamperometric titration sets it apart from the rest. Biamperometric titration, a testament to the versatility of amperometry.